All right, so you may recognize some of these guys as scientists. All of these people are scientists, but I'm sure there's some you don't recognize. So we're going to talk about the fundamentals of biology. Specifically, we're going to kind of scale it back and talk simply about science today and the scientific method. Now, you may know this guy on the top left and recognize his portrait. That is Charles Darwin, the um, person who published a very lengthy work discussing his theory of evolution, which has completely changed how we do biology today. You're probably more familiar with the guy on the bottom right, Bill Nye, the science guy, who is somewhat of a scientist, but more of a public figure in trying to help others understand science and use science in our world today. The top right you may recognize as Watson and Crick, the scientists who discovered the structure of DNA, which relates to its function and allows us to do many things in science. In the bottom left, this is probably the most famous current living biologist today. And my guess is most of you, probably 99% of you, don't know who this is. And his name is E.O. Wilson. He's a sociobiologist, has done a lot of work with uh, ant and social behavior of animals, and then kind of extrapolating that to, well, what does that mean for our society? So a very influential person in biology today. We're going to go through, well, how do scientists do science, right? Well, first let's do a definition of science. And if I give you a few minutes at home to define science in your own words, what would you say? And when I do this in class, generally people talk about the scientific method. They talk about a way of thinking and organizing facts. My definition is the formulation and or discrediting of objective explanations for the observation of phenomena of the physical and biological universe. This is also not even a comprehensive definition because math is a science, but it doesn't fit within this scientific definition. But the science that I do as a biologist, or I'm involved in understanding and teaching, where it has a grounds in experimentation and building off of experiments, this would fit. What does science depend on then? Well, it depends on us, depends on data, depends on taking information and figuring out what to do with it. So data are measurements that we can accumulate and store and then try and make sense of. So we can break data or objective measurements into two different types: quantitative and continuous variables or measurements, and qualitative or grouping measurements. So the types of bears would be a qualitative measurement. Male and female, we're putting individuals into two groups, so that would be qualitative. Temperature, however, is continuous, meaning it is on a numbering system, goes from um, 0 or below 0 to infinity in either direction. And it can be broken down into decimals. So that would be a quantitative measurement. Light can be broken down into different wavelengths, and so it can be quantitative, but it can also be qualitative. So if we just say red, green, yellow, blue, that's grouping them into different colors. How do we make objective measurements? Well, we use tools. We have a few tools here. A thermometer is a tool, a ruler is a tool. There are other tools which extend our measurements beyond our senses, including um, microscopes and voltage meters and EKGs. All of these things allow us to do more with our senses than what we can do um, on our own. So once we then have data, one thing we can do is we can try and manipulate our surroundings to where we can produce data which help us answer a question. That essentially is the scientific method. The first thing that we do, though, is we make observations. We collect that data. We look at, we have observational research. We try and understand these observations through logic, including deductive and inductive reasoning. In deductive reasoning, we take general information and apply it to a specific situation. So Sherlock Holmes is deducing, right? He knows things because he has recognized all these general information patterns. Inductive is the opposite, it's where we take specific information and apply it generally. So we know that motion of different objects will behave in specific ways because we have theories and laws which help us understand them. So once we have kind of worked through our logic, we can then ask a question and form a hypothesis as to why those things are occurring. So, for example, marine populations are significantly increasing, decreasing, not changing, and unknown in different percentages. This is based on a study done in 2013. What are some of the questions or hypotheses you might have about these data? Well, it might be, since most of them are significantly increasing, we might want to know, well, why? How come in the world where most other things are decreasing in population number because of human influence, why are these increasing? So our next step would be then to test or design an experiment or go out and collect data in a certain way where we can answer that question. So we aren't going to continue with the whale example or the marine mammals example, but we'll do something more simply. In order to do an experiment, we first need to identify variables, which are things which are going to affect the outcome and can, in an experiment, be manipulated. The independent variable is the one that we are going to manipulate, and we are going to know what the amount is or what the level is. And the dependent variable is going to be what we measure, what the outcome is. So in our example down here, we have two seeds, two pots, which we're going to plant. One we're going to water and one we're not going to water. And our question is, does watering help plants grow? So our independent variable is the amount of water. And we're going to do a fixed amount every day, and the other one we're not going to do any. So it's really a categorical variable. We have watered and unwatered. The dependent variable is going to be the number of seeds that sprout. And what we found was, in the end, 9 out of 10 seeds sprouted when we watered them, and 0 out of 10 sprouted when we didn't. In our experiment, we can have positive and negative controls. So this is a very basic experiment, but let's say we wanted to add then a level of fertilizer. So we're going to take our two pots. One will have fertilizer and one won't. And we have a positive control, something where we know something's going to happen. Because we've done this experiment, we know that water is going to cause sprouts to grow. And then we would compare water plus fertilizer to just water to determine the effect of fertilizer. Our negative control would be where we know that we're not going to have anything. So we could also grow peas without any water, and we know nothing's going to happen. So that would be our negative control. All right, there are different types of experiments and studies. So not all studies are experiments. Some of them can be purely observational or just collecting data. Okay, so we may be studying the migration route of gray whales, which is the top right picture. Well, that would be just something that's observational. We just want to see where they're going and how far and where they stop and things like that. A controlled experiment is what we did with the plants. Or in this bottom left here, we could do different experiments with the ability of cetaceans like dolphins to interact with some sort of thing which we put out for them. We can have quasi-experimental experiments where we control some things but not everything. So this is generally done in the field. So let's say we have two plots right next to them. One of them we're going to put a bunch of corn out there, and we're going to see how many deer come to that one compared to the one where there's no corn. Well, it's not fully experimental because we don't control all the variables. We just are controlling the amount of corn. So quasi means partly. And then opportunistic studies. So let's say we have a fire that rolls through a forest, and we want to look at the deer populations after a fire. Well, we didn't start the fire, we didn't manipulate the environment, but we're going to take advantage of this fire and use it to study deer populations in response to fire. All right, so we'll look at this in class, but this is a video of a whale fall and researchers then going and just looking at the different composition of animals and species on this whale as it fell to the bottom of the ocean. And this is a dead whale, right? So scientists have actually done this. They'll take a whale that's washed ashore and they'll bring it out to the middle of the ocean, they'll let it drop, and then they'll go down and look at it and check on it a number of times and look at the different animals that are feeding off of it at different times. So this is an observational study. It's a little bit opportunistic because we took advantage of the death of a whale, and sometimes you can find whales that have naturally died, and you would go and study those. But it's not, we haven't manipulated anything. We're just taking advantage of opportunities to observe something. So on the other hand, let's look at some data, some observations, and do an experiment. So dolphins eat fish. Fish have mechanisms to avoid being eaten, and counter shading helps fish avoid being eaten by dolphins. So counter shading is where you have a dark top of the fish and a light underbelly of the fish. And the idea is predators looking from above can't see the fish because they'll be blinded by the light, which will be the same color as the fish, and predators looking from above will see the darkness below them, and the fish will be camouflaged with that. So let's say we have some dolphins. We're going to look at variables, observations, and hypotheses with the information we have. So we want to know if dolphins can catch fish that are counter shaded versus those that aren't, and to see if they can avoid our predator, our dolphin, better or worse. And that would be our experiment. So we need a control though. So we could have our regular fish, which is counter shaded, as our control. That's how they naturally are. And then we could paint those fish so that they're all dark, and then we can paint other fish so they're all light, and then compare it to our control, our natural one, to see what the outcome is. Well, in the end, we're going to get some results with our data, and we often want to display this results in a way that other people can see and make sense of it, and so that's often a graph or a chart. We usually use the x-axis to represent our independent variable, and our y-axis our dependent variable. A histogram measures the frequency of different groups, and a pie chart is not used too often in science, but it just shows proportions of different variables. Statistics are mathematical comparisons of data. We're just going to briefly touch on those, but they're generally based on probabilities, the probability of achieving your result as compared to randomness or chance. We have descriptive statistics. Those would be like the mean or the average, the median, mode, range, and standard deviation. And we have inferential, where we are trying to understand the relationship between two groups or two variables. That would be a t-test and a correlation. So if we do our dolphin experiment, and these are the times it took for the dolphins to catch the fish that were normal, and then we have our other group, which was our painted fish. Okay, the first thing we would do is our descriptive statistics. So we'll calculate the average, median, mode and range for each. Oh, and then we would look at the statistics. So here's our range is 34 and 44. Our mean is 39 seconds and 45 seconds, and our standard deviation is, oh wait, no, so these are off a little bit. N is 32, N is 33, range is 34 and 44, minimum is 5, max is 39, the mean, the average is 15.3, and standard deviation is 8.8. Now you need those in order to run a inferential statistic called a t-test, and that gives us a p-value. Here the p-value is 0.681. What that means is that 68% of the time our results would be similar to randomness, which isn't very different, right? So you're saying if I were to just randomly grab samples of fish, our results aren't much different than those random results. So what do we conclude? Well, we can say there really isn't evidence that counter shading with this fish helped in the avoiding of these dolphins. Alternative hypothesis might be, well, maybe it's the speed of the fish, and because all these fish were the same, they didn't have a difference. Can we say that counter shading does or does not help with predator avoidance? Well, not generally, but we can say with this one fish, with this one experiment, and these dolphins, they did not support that theory. All right, and that would be our conclusion, the interpretation of the results. So in that instance, we would say this hypothesis maybe needs more data. Maybe we need to do lots of different types of fish. Maybe we need lots of different types of dolphins. Maybe we need to do it in the wide ocean rather than in a tank. Maybe that would affect it. And so when we talk about conclusions, we're talking about what we did, what we found, what we can say is specific, and the other things we could have done differently. All right, last part of the scientific method is validation, which is, can your experiment be done over and over again with the same results? And the more times you do it with the more subjects, the more confident you can be in your results. Now we had a good sample size. It was about 30. It was 33, right? But we could be more confident if we had 300. Right? So increasing the sample size increases the confidence of your results. All right, finally, we're going to look at this little case study here. This is data that I have I collected for my dissertation, where I looked at the length of these whales and the calving interval, which is the years between pregnancy events. So, for example, 00B5 was 17.5 meters long and had a calf, or was pregnant, every 3.225 years. Okay, and my question was, is there a correlation between length and calving interval? And when you look at the graph, this is what it looks like. So the pregnancy rate, how often they were pregnant per year, and how long the whale was. You can't really see it, but the p-value is 0.170. And we would say we want something below 0.05, so below a 5% chance. So we would say this is not significant, not supported. However, if we look at the age data, we find that you can see it looks a lot more correlated. So the older you are, the shorter your time between pregnancies. And the p-value is 0.001. That means there's a 0.1% chance to get these results compared to randomness. And so we would reject the hypothesis that there's no correlation between these two and say we have evidence that suggests that the older you are, the more frequent you can have babies if you are an adult bowhead whale. Okay, so those are some of the scientific method and things that are important to know, and we will build off in class.